Thank you so much for the introduction, and it's wonderful to be here in person at an in-person meeting discussing this really important topic. So I'd really like to thank the organisers for giving me the opportunity to talk today, really about what we know about how a suboptimal in utero environment impacts on our long-term cardiometabolic health, and how really by understanding those mechanisms, that can give us rational insight to develop key intervention strategies to improve the health of women and their children. So as we heard in the wonderful keynote lecture this morning, this is embodied in what's been called the developmental origins of health and disease, or developmental programming. So the idea that if we're exposed to a suboptimal in utero environment, that has structural, cellular, and molecular, including epigenetic changes that impact on our long-term health, which include increased risk of cardiometabolic disease. And this field really started by those pioneering studies of David Barker, focusing on undernutrition. But since then, we really recognize that it's not just undernutrition in utero that impacts on our long-term health. There's a whole range of other factors, not just nutritional, but includes stress, hypoxia, air pollutants, maternal smoking, and the list is growing. For the purposes of today's talk, I'm going to focus on a nutritional suboptimal environment, that of overnutrition. And this is particularly relevant in both the developed and developing world, because we know that obesity rates are increasing across the globe. And this was really highlighted by a recent report by the Chief Medical Officer of England, who actually highlighted in the UK now, over half of pregnant women are either obese or overweight. So if you're a lean pregnant woman in the UK, you're in the minority. Now that's of concern because we know that obesity during pregnancy in itself has immediate detrimental consequences for both mother and child, including increased risk of developing gestational diabetes, and that's thought to contribute to why now 1 in 7 pregnancies globally experiences gestational diabetes, with a range of other immediate consequences for mother and child, including increased risk of preeclampsia. Now, as well as these immediate detrimental consequences to mother and child, there's growing evidence to suggest that children born to an obese mother are themselves at increased risk of poor cardiometabolic health, and that this transmission of poor health from mother to child doesn't simply reflect transmission of obesogenic genes from mother to child, but is more epigenetic in origin. Now, in terms of evidence from human studies that developing in utero in an obesogenic environment impacts on our long-term health, really that comes from studies such as this one here, that was carried out by a group in Canada several years ago now, looking at children living in the same household, so that's controlling for current environment as much as possible, born to the same parents, so controlling for genotype as much as possible, but born either before or after the mother had weight-reducing bariatric surgery. So as you can see on this slide here, the child born after maternal bariatric surgery, i.e. when the mother was leaner, is themselves leaner, has a lower insulin and a lower blood pressure than their brother or sister who was born when the mother was more obese. So this is certainly consistent with the fact that developing in utero in the face of overnutrition impacts on our long-term health. However, in all human studies such as this one, it's really difficult to remove all confounding factors and even more difficult to try and tease out underlying molecular mechanisms. So this is where animal models are very important, because firstly, they can help address whether these relationships are indeed causal, and they can also help us explore the underlying mechanisms. So in my laboratory, we use this mouse model of maternal diet-induced obesity, where mice are fed either a control diet or this high-fat diet supplemented with condensed milk for 10 weeks prior to pregnancy. So these dams are obese when they enter pregnancy, and this is primarily due to an increase in fat mass. They remain on this diet throughout pregnancy and lactation, so they remain obese throughout this period, and as you can see at weaning, on this bottom panel here, the moms are still heavier and considerably fatter than those fed the chow diet. In most of the data that I'm going to show you, the offspring themselves have been weaned ad libitum onto a healthy, low-fat chow diet, so the effects that we're looking at is due to this exposure to maternal obesity. So what is the consequence of developing in utero and being suckled by these obese dams? So if we firstly simply look at body weight of offspring weaned onto a chow diet, the red line is the offspring of the obese dams, the yellow line the offspring of the lean dams. So you can see that over the first few months of life, the offspring of the obese dams have a very similar weight to the offspring of the lean dams when they're fed a low-fat diet. However, if you wean these animals onto an obesogenic diet, more reflective of the environment that we experience today, you can see that both groups of offspring gain excess weight, but those that were exposed to maternal obesity are substantially heavier than those who were born to lean dams. So they're more susceptible to excess weight gain in the same obesogenic environment. Just like in their mother's case, this excess weight gain is related to an increased adiposity. As you can see, those offspring that were exposed to maternal obesity are really primed for excess weight gain. This excess weight gain is preceded by an increase in food intake, and so we think this is related to programmed changes in the hypothalamus, in particular the arcuate nucleus. It's related to early insulin resistance that actually we can even detect in the fetal hypothalamus, and we believe it's related to programmed changes in microRNAs. In the interest of time, I'm not going to show you all that data now, but I'm very happy to chat more about it later on. What I want to focus on more is these offspring who've been weaned on to a chow diet. Because really, at first glance, you may say, well, so what about developmental programming? If I myself eat a healthy diet, then it looks like my body weight is going to be okay. But just because body weight is okay, that doesn't mean to say these animals are metabolically healthy. And so we've looked at these animals at 8 weeks of age, when there's absolutely no difference in body weight and no difference in adiposity. And one of the earliest differences that we see is in the heart. So despite having a normal body weight, these animals have increased heart weight. We've done an analysis to show that it's a left ventricular cardiac hypertrophy with an increase in cell size. This hypertrophy is pathological in nature because we show induction of fetal genes associated with pathological cardiac hypertrophy. As well as these structural differences, we also see functional differences in the heart, as shown on this next slide here, where we've done some working heart Langendorf analysis. And as you can see, the animals show both systolic dysfunction, so a reduction in left ventricular developed pressure, as well as diastolic dysfunction with an increase in left ventricular end diastolic pressure. So for any cardiologists amongst you, you will know that this is really reflective of a struggling heart trying to maintain cardiac output and likely to be on the pathway to heart failure. As well as these effects on the heart, we also see effects in metabolism. So at 8 weeks of age, the offspring of these obese dams, despite being normal glycemic, need to produce more insulin to maintain glycemia. We've recently done euglycemic hyperinsulinemic clamps to show that this hyperinsulinemia is indeed reflective of whole body insulin resistance. So these studies show you that there's a causal relationship between obesity during pregnancy and offspring cardiometabolic health. So clearly, particularly in light of the growing prevalence of obesity, the next question is, well, what can we do about it? How can we intervene and prevent this growing pandemic of obesity? So clearly, in an ideal world, we'd have all women entering pregnancy with a healthy BMI. But as we all know, we don't live in a perfect world. And actually, if anything, prevalence of obesity is getting worse. And that was highlighted by recent statistics released in the UK, looking at 11-year-old children and their adiposity and how it's changed since the start of the pandemic to now. And there's been an almost 5% increase in obesity in those children. So quite worrying, because they are obviously going to be the parents of tomorrow. So really we need to act now to think about, well, how do we prevent this transmission of poor cardiometabolic health in the mother to the health of the child? And so clearly we want to intervene as early as possible, which means intervening during pregnancy, with the hope of improving maternal health and therefore consequently that of our child. But it's really difficult to think about intervention strategies if we don't understand the mechanisms and don't know what it is about that maternal obesogenic environment which is mediating those long-term effects in the offspring. What is it about that obese mother which is driving those effects on the fetus? What are those epigenetic drivers that are leading to that programmed poor cardiometabolic health? And so once we know that, then we can start thinking about designing intervention strategies which target those factors associated with obesity, which are imprinting, in essence, those effects on the fetus. And so clearly we know that obesity is associated with a number of different factors. As I've mentioned, obesity is associated with increased risk of gestational diabetes, so perhaps it's the glucose driving the effects in the offspring. We also know that it's a state of insulin resistance. Maybe it's the hyperinsulinemia driving those effects in the offspring. We also know that it's a state of lipotoxicity, so maybe it's the lipids which are driving those effects on that developing fetus. It's also a state of inflammation and oxidative stress. Perhaps they're the driving factors. And obviously, let's not forget the placenta, because that is obviously the interface between mother and child. We also have to recognize that it might not be anything to do with the maternal metabolic milieu in itself, but may be related to differences in maternal dietary intake and perhaps maternal weight gain. So we wondered whether we could use our mouse model as a tool to really start dissecting out which of these factors could be programming those long-term effects in the offspring. And we did something very crude and just looked at the various parameters in the mother and looked to see which were most strongly correlated with, in the first instance, offspring hyperinsulinemia. And as you can see on this slide, the data was actually quite striking. So we saw this positive correlation between maternal insulin and offspring insulin, yet there was no significant correlation between maternal adiposity per se, or maternal body weight, or maternal cholesterol, leptin, triglycerides, or free fatty acids. So this may have a very important translational message, because it may suggest that we don't need to focus on the adiposity per se, but focus much more on improving maternal metabolic health in relation to her glucose-insulin axes, and try and look at ways of improving maternal insulin sensitivity. Now, obviously, this is just a correlation and doesn't necessarily mean that it's a causative relationship. So to start addressing that, what we need to do is look at ways of improving maternal insulin sensitivity and then look to see what that does to the offspring. Now, there's a number of approaches that we could take, but in the first instance, we took a physical activity intervention and did some experiments using a treadmill training regime. So again, we have our control and obese group of dams, but this time we introduced this third group of animals where 1 week prior to pregnancy, they were initiated on this treadmill training regime, such that by the time they entered pregnancy, they were happily doing 20 minutes a day on the treadmill, 5 days a week. No prizes for guessing the rationale for this study design. When you have this study design, this means that the mice, the students and the postdocs all get the weekends off, so perhaps in itself also more translational. So why we did it 5 days a week and not every day of the week. So as we heard earlier, the gestation period in a mouse is around 21 days, and so we continued the exercise intervention up until E17 of pregnancy, and then firstly looked at the effects on the mother on E18 of pregnancy. So if we firstly look at her glucose tolerance, as I mentioned before, obesity is associated with impaired glucose tolerance during pregnancy. This short exercise intervention was able to completely prevent that development of impaired glucose tolerance. Likewise, when we looked at the insulin axes, as I showed you before, the maternal obesity is associated with hyperinsulinemia in the mother. That is again prevented by the maternal exercise intervention. Now, importantly, this improvement in maternal metabolic health is not associated with DAM weight loss. And I don't think anybody would advise a woman to lose weight during pregnancy. And so as you can see, weight is maintained, I think, because it's a short period of exercise intervention. And throughout this, the DAMs are being fed ad libitum. They also maintain their adiposity. So as you can see, the obese exercise DAMs have an equivalent adiposity to the non-exercise DAMs, which is significantly higher than that in the controls. So essentially what we've done is converted this obese, metabolically unhealthy DAM to this, if you like, fat-fit DAM. As you would expect with this degree of adiposity, the DAMs are also hyperleptinemic, which will also help us tease out whether any of the programming effects are driven by leptin. So what are the effects on the offspring? So firstly, if we look at the glucose-insulin axes, we can see that the maternal exercise intervention, as well as improving maternal insulin sensitivity, at 8 weeks of age, which is a young adult for a mouse, it's also prevented the development of insulin resistance in the offspring. One of the epigenetic changes that we see relates to a reduction in insulin receptor substrate 1 in adipose tissue. The adipose tissue seems to be a key site of insulin resistance, which is very vulnerable to a suboptimal early environment. And as you can see, that programmed reduction in IRS1, which is related to programmed increase in expression of MAY126, is also prevented by the exercise intervention. Likewise, when we look at the heart rate, we can see that the increased heart rate is prevented by the maternal exercise intervention. Likewise, the functional defect, in this case the reduction in ejection fraction, which we measured by echocardiography, is also prevented by the exercise intervention. So this all looks very positive. Now, the other aspect of programmed phenotype that we see is an increase in blood pressure. And we were completely expecting, based on the improvement in cardiac function, a normalization of offspring blood pressure. But indeed, when we looked at blood pressure, that is not what we saw. These animals remain hypertensive when the DAMs had been exercised. So this is important for a number of reasons. So firstly, it suggests that the mechanistic bases mediating the effects of maternal obesity on offspring hypertension are not the same as those that are mediating the effects on cardiac dysfunction, which means you're going to probably need separate intervention strategies to prevent both of those outcomes. And so it would also suggest that it's not insulin which is mediating these effects on offspring blood pressure. And actually, when we went back to the maternal data, we could see that indeed there was no correlation between maternal insulin and offspring blood pressure, suggesting it's another factor which is mediating these effects. What that factor is, we don't know. It could potentially be leptin. As I showed you, leptin is not normalized by the maternal exercise intervention, and there's data out there to suggest that certainly neonatal hyperleptinemia is associated with programmed hypertension. So this in itself suggests that we need a separate intervention which is going to prevent the offspring blood pressure. Now, in the context of human gestational diabetes, in many populations, including the UK, when lifestyle interventions fail, the first-line pharmacological therapy is metformin treatment. So metformin is widely used in the UK to treat gestational diabetes. and is tolerated well by many women. And so following on from this data with our exercise intervention, the next intervention that we looked at was a maternal metformin treatment intervention. So just like in the case of the exercise intervention, we initiated this just prior to pregnancy and maintained the supplementation with a clinically relevant dose of metformin up until the end of gestation. And so firstly, looking at the effects on the mother of this metformin treatment, there's large amounts of data in the human literature looking at very many different beneficial effects of maternal metformin treatment, one of which is an apparent reduction in gestational weight gain. We replicated that completely in the mouse model, where we see that the metformin treatment is associated with a reduction in maternal fat mass. Now that's clearly beneficial for the mother going into any subsequent pregnancy, because it means that she's going to end this first pregnancy at a leaner state than she would be if she hadn't been on metformin, which should be beneficial for her health, as well as the offspring born to a subsequent pregnancy. As well as a reduction in total body fat, these mothers also have a reduction in liver fat, so again, very beneficial. Also see beneficial effects on vascular function in the mother, and this is again consistent with data in the human literature to suggest beneficial effects of metformin in relation to preeclampsia. So this all looks very good in terms of maternal health and her health at the end when she enters any subsequent pregnancy. However, we've also started to look at the offspring. And this is again, as we also heard in the last presentation, all the data I've shown you to date is in male offspring, but we've also started doing more and more studies in both males and females, because actually one of the advantages of a mouse model is you can look at littermate siblings of the two different sexes, so it's really controlling for fetal environment. And when we look at the offspring of these dams that have been treated with metformin, what we see is firstly nothing particularly beneficial, because actually rather than improving offspring adiposity, specifically in the male offspring, the metformin treatment seems to increase adiposity in that exposed group. Now, metformin freely crosses the placenta, and so you get equivalent concentrations in the maternal circulation and the fetal circulation. So we suspect this may be related to direct effects of metformin on that developing fetus. You don't see that effect on adiposity in the females, certainly at 8 weeks of age. As well as causing increased adiposity in the males, if you look at, if you like, the metabolic healthiness of this fat, we see that not only is there more fat, it's more inflamed, so it's metabolically unhealthy fat. And again, you don't see that at all in the females. So the response to that treatment of the mother with a drug, which freely crosses the placenta, is sexually dimorphic in terms of what it does to male and female offspring. Now, clearly, if you're giving a drug during pregnancy with the hope of improving both maternal and offspring health, it's not particularly beneficial if you've got evidence for increased adiposity. Now, clearly, these studies are in mice, and a mouse is not a human. But when we find data like this, obviously it's really important for us to look to see whether there's any evidence at all that this also happens in the human context. So to address this, what we've recently done is a systematic review and meta-analyses looking at all the published randomized controlled trials for treatment of gestational diabetes, where they've compared insulin treatment with metformin treatment. In each case, glycemia is equal, because obviously if metformin doesn't normalize maternal glucose, then they're giving additional treatment. Now, when you look at that data, the first thing to note is actually there's very little follow-up data, but there's a lot of fetal growth data. And so there was 28 studies in all, and we could see that metformin treatment during pregnancy was associated with a significant reduction in birth weight. As I mentioned, there were very few studies which had followed up those offspring. However, the 3 that did exist showed that although the offspring was smaller at birth, in infancy they underwent catch-up growth and were heavier, and by the time they were at primary school, they were significantly fatter, and almost a 1 BMI unit increase, which is actually quite clinically significant. Now, unfortunately, none of these studies were powered to look at any sexual dimorphism, so we don't know whether from the human data there's any evidence to suggest that there's also sexual dimorphism operating there as well. So, in conclusion, I hope I've convinced you, if you needed convincing, that obesity during pregnancy impacts on both maternal and offspring cardiometabolic health. We've got wonderful mechanistic insight from animal models which show not only that there's a causal relationship, but start to help address underlying molecular mechanisms, and one of these seems to be the important role of maternal hyperinsulinemia in driving these effects in the offspring. And so clearly that gives us insight into potential intervention strategies, and insulin is a potential target in the mother of intervention strategies. And so I've also shown you that there can be beneficial effects of some interventions on offspring outcomes. But I hope what I've also shown you highlights that not only do we need to consider the effects of any interventions during pregnancy on the mother, we also need to consider the effects in the offspring, and it's particularly relevant to what we're discussing today. We also really need to consider fetal sex, which may not respond in the same way to the same fetal environment. And so what I also would like to highlight is also the need for long-term follow-up studies in both humans and in animal models, and again, making sure those studies are powered to allow us to statistically look for differential effects in males and females. And in relation to this sexual dimorphism, I think actually, although it's a really underexplored area in the context of developmental programming, it's actually a really important area, and there's a lot of evidence out there to suggest, A, that there will be sexual dimorphism in responses to the suboptimal environment in utero, and actually also there's some actually good indicators of what some of those mechanisms will be. And actually I've just done a very thorough review of this topic, which will be coming out in Nature Metabolism next month. And I just want to highlight some of those potential factors which could explain why there is dimorphism, sexual dimorphism in response to the suboptimal in utero environment. It may again make us think more back to this question of precision medicine, and should there be different treatments for different types of suboptimal in utero environment which are mediated by differences in sex. Now, these differences are not just going to be mediated by differences in sex hormones. There's actually a lot of knowledge out there already about sex differences in some of these developmental processes. So, for example, a male and female placenta are very different. There's a growing number of studies out there, we've got our own data as well, to suggest that their transcriptome is different, their lipidome is different. There's published data from humans, we've got our own data in mice, and in the mice, as I said, these are littermate siblings, so this is inherent differences in the placenta when they're exposed to the same in utero environment. We also know that female placentas are actually more efficient than male placentas, which could explain some of the vulnerability of a male to a suboptimal in utero environment. We also know that male fetuses grow more rapidly in utero compared to females, so again, maybe why they're harder hit if they're exposed to anything which is reducing fetal growth, which, as I showed you, could be metformin, could fall into that category. We also know that males and females age very differently. I'm sure we'll hear more tomorrow about telomeres and sex differences in telomeres. There's data for sex differences in telomeres in humans. We have our own data in rats. We think accelerated aging is actually a key mechanistic part of this whole concept of developmental programming, because many of the conditions associated with a suboptimal in utero environment are those that people traditionally develop with age. And so what some of these things may be mediated by is accelerated aging at the cellular level. For example, we see premature induction of P16 as a consequence of low birth weight and catch-up growth in the rats. That's associated with accelerated telomere shortening. Also differences in lifespan, with again, the male rats having a shorter lifespan, as you see in humans, than females. So we've got a lot of potential areas to follow up that based on existing data. And so I think this is definitely an area where we're going to hear much, much more about in the next few years. And so my last, but probably most important slide is my acknowledgement slide. I'm really just a spokesperson for a lab of very talented postdoc students and technicians. So I really have to thank them for their enthusiasm and passion for this field. Whenever I give a talk, I always want to highlight as well is that this is a field that really crosses so many different disciplines and is not a field that any one lab alone is going to be able to solve. And so collaboration, more than anything in this field, is critically important if we really want to take it forward. So I always encourage people to collaborate widely, share data early, and then we can really make a difference and an impact. And obviously last, but by no means least, the funding bodies, because as we all know, without them, none of this is possible. Thank you very much.